Good morning. Good morning, everybody. So today we have a coffee break with TRANSFAC again. So grab your coffee like I do. Okay, so today we have a very nice topic. So I will share my screen. It's called what is away from transcription factors to pathways. So let me first put it in presentation mode. So as you know, my name is Alexander Kell. So geneXplain company, and we are developing TRANSFAC database. It's quite known and well known, I would say. So if you are here, probably you heard about TRANSFAC. So it's a database about transcription factors and their binding sites in genome. And today we will touch that point that actually transcription factors, they are not functioning alone. Somewhere in the cell, they are in some network, in some pathways. And to find a way from transcription factors to pathways, and actually even to try to find what is a master regulators in those pathways which control those transcription factors. And that's the topic of today. And actually, as usual, you are welcome to write any questions. We have here in Zoom Q&A section where you can click and type your question. I will look if some question appears during my talk, and maybe you can even interrupt and answer your questions. Also, at the end, we'll have enough time to answer all your questions. And I also will come to the questions we got so far in between our coffee breaks, and we'll try to answer them. Okay, so what is away from transcription factors to pathways? Well, the first thing we have to understand that transcription factors, they act in the nucleus, and basically they bind to the regulatory regions of genes, and they regulate the expression of those genes, which is measured by transcriptomics usually. So when we do RNA-seq, for instance, we know which genes are upregulated or downregulated. And those genes may encode, for instance, enzymes, which do some metabolism of the cell, or they can do some other things, and we will see what kind of things they can do. As I said, in the promoters of those genes, also in enhancers and other regulatory regions of those genes, we have target sites, and which are bound by transcription factors. And in fact, those binding sites can be also approximated by various, ChIP-seq epigenomic methods, and we will not talk about that today. We had it before, but you are welcome to ask about these questions, how we can use epigenomics data to find better our binding sites. Today the most focus is on what happens after we found transcription factors. So we identified transcription factors, or we predicted these are transcription factors which might be responsible for upregulation of my genes. And then I ask a question: these transcription factors are endpoints of some signal transduction cascade. So as you know, and I will show it today a bit in examples. These factors to be active, it's not enough just for the protein, for these proteins, or even sometimes complexes of proteins to be expressed. It's not enough. They have to be activated. So which means sometimes they have to be phosphorylated, or some other kind of modifications should happen that they become active to bind to their binding sites and to regulate genes. And they are activated by action of other components of regulatory machinery, which are called kinases in case of phosphorylation, and kinases, which are in turn are also activated through a signal transduction cascade, which can be actually found in some proteomics experiments. So basically, those components of signal transduction cascades, these are kinases or other proteins which play a regulatory role. These are proteins, and you can detect them in the various experiments. Also, when you don't detect them, and we can assume they are acting there, we can get the majority of that data about signal transduction cascades from databases. And I mentioned in my list like TRANSPATH database, and I will show today. Also, REACTOME database has a number of signal transduction cascades, and also can be any protein-protein interaction databases which can contribute to this information. And at the top, we can come up with a kind of, we call it master regulator. So maybe a few of them, some important proteins, so the accomplices which after being activated, for instance, if it's a receptor in the membrane of a cell which got a ligand and activated. So this one receptor can trigger the whole cascade of events, which leads to activation of a number of transcription factors, not just one, but really a number, which in turn will try to regulate, will manage to regulate those genes. All right, so that's how the overall schema of regulation of the genes and the way how these transcription factors are connected to it is represented here. But of course, as usual, devil is in details. Let's go into the details. So first, and that's I talked about that before. So we predict the binding sites in promoters of the genes. So, and for that we use positional weight matrices, which are collected in TRANSFAC, and those matrices are built on the basis of known sites, so which are collected during our curation process. And then when we have those matrices and we have them, we have TRANSFAC is the biggest collection of matrices. So we have for vertebrates, we have more than 7,000 positional weight matrices. And recently we were counting how many factors actually have at least one positional weight matrix in TRANSFAC. And we found out that more than 93% of all known transcription factors which can bind to DNA in human actually have at least one PWM positional weight matrix in TRANSFAC. So it's a pretty good coverage of transcription factors. But still, we have the motif, we have the positional weight matrix, but we have very little knowledge where those sites can be, where those factors can bind. So some knowledge comes from RNA-seq, but it's far not being enough because of various conditions where those binding can happen. And so we have to predict those binding sites. So, and we do it with some matrices, with some scores. We call it match score, and that's the algorithm called match, which is able to predict binding sites using the scoring function. And then also I was talking about that a lot. Then basically those transcription factors, they are not usually acting alone. They are acting in complexes and to activate a certain gene. For instance, in this case, through the enhancer. The genes, this interferon beta of human is activated when in a particular situation, these 5 transcription factors are got activated, and then they bound together in synergistic manner, and they activate the gene. And we actually had developed tools to find those complexes, and important to know that to understand that those complexes of factors like these guys, they are very fuzzy. So they can bind to the DNA sites, not very precise, and they bind to each other, like I showed in the previous picture. And by doing that, they stabilize this complex and they activate the gene. But in another condition, when another repressor, for instance, can come here in a very active mode, and it can basically remove all those factors and silence the gene, although the DNA sequence is the same. So depending on condition, which factors are activated, which are not activated, you see a different regulation of genes. And that's again to emphasize that this cascade of regulation of activity of transcription factors is really, really important to understand how genes are activated. All right, so we had this tool, which is called Composite Module Analyst, and we have them in our offering in TRANSFAC associated platform. And this tool, what it's doing, it's taking promoters of the genes here at the start of transcription start site, and the first exon of the gene is started from here. But in the promoter of the gene, the algorithm scans a window, and it searches for the combinations of binding sites for transcription factors, taking into account all possible combinations and finding those clusters of binding sites. And they will see that in the tool. And basically, and again, once we found those transcription factors in combination which are acting in my situation, in my experiment, then the question is, what is a master regulator? And maybe this master regulator is important to find because we want to control those expressions by some drugs. That's another story. But still, the major idea is really try to find these master regulators. And how we do that? Basically, let me first show this picture. And that's a major topic of today. So to explain you how this master regulator search is acting and how we actually connect transcription factors, which are blue dots here, to the whole network of signal transduction, and basically at the end find the master regulators. So this network comes from TransPath database, which is a database on reactions mainly. So we have more than 1 million signal transduction reactions collected over many years from literature, where actually those reactions are described in very precise details, like this, for instance, this kinase is phosphorylating this kinase in a particular situation, and this kinase is phosphorylating this transcription factor in another situation. So basically, by putting this all together, we built such a reference network. Of course, this reference network is never working as a whole in any cell. It's very virtual. It's a reference because, as I mentioned, all these reactions may happen in different conditions. But in a particular situation, in particular condition, only a certain part of that network is actually acting, depending which factors are present in the cell, which kinases are present and active in the cell. And basically, we have to find a way through this reference network to the master regulators. And this is done with this kind of formula. So basically what we do, we do the search for shortest passes in this network. There's an algorithm to search for shortest pass, which means minimum number of steps from one molecule to another molecule. It's similar to the search for shortest pass in your navigation system when you drive the car. So basically, that's exactly the same algorithm which is working here, except we in the car usually have one point to start and another point to end. Here we actually have several points to start and several points to end. So it's like not just one car, but it's like a run of many cars, and who comes first? Like on a run, on a competition run between different cars. And here, who will win is actually who will come to the end first, and that is calculated by this formula, which is actually the following. So, for instance, if we take this molecule, we see that through this cascade, the signal can reach all of those transcription factors, right? So it has a lot of reach. But this molecule, if you activate it, it can reach only these 4 because it doesn't have other connections. So in one sense, maybe this is less important than that one. But you have to take into account that this one can be really a hub. So which means a kinase or a certain regulatory protein, which acting in practically all types of cells. So it really can, it's a hub of connecting all signaling through it, and it activates a lot of transcription factors, but it's not specific enough. So basically, if we are saying only these 6 transcription factors are important, and others are not important, so then this hub may be not very interesting, because it will activate not only these factors, but it activates all other factors as well. That's why in this formula, M is actually number of transcription factors which can be reached through these connections from that network, from that molecule. And in the below here is actually a number of other transcription factors, which are actually this N, which can be reached in general. And actually, if it's a big number, the whole score of that molecule goes down. So in that competition, actually, this one wins. Because actually, although it covers small amount of transcription factors from my list, but it's very specific in general. It doesn't cover many other factors, and that's why the score of that molecule gets higher. All right, so that's how the general algorithm works. I need a slip of water. And but actually, there's another very important variant of that algorithm, which takes into account the amount or activity of the proteins which stands in between. So those connecting proteins, as well as transcription factors as well. So, and this data may come from outside. For instance, by doing proteomics experiments or phosphoproteomics experiments, you have a list of proteins which are active in your cell. So you can put this list in the algorithm, and what will happen, these proteins will be mapped to the whole reference network, and then those ones which have a higher value, higher activity from your experiment will get a higher weight in this weighted graph. And then actually the search will be done in the weighted graph, which means those connections will be sort of shorter. And it's again like in a navigation system, it may happen that the longer path through the highway, in fact, in time-wise, it will be faster, although it's long in a distance, but it's faster in the time. The same is here. Maybe number of steps can be bigger, but the steps are shorter because this protein was found in the experiment, and we actually attract the algorithm to those points by putting weights on that graph. And in that case, we may find another master regulator, which is more appropriate and more correct, because we know which component of this signal transduction network are actually working more actively. All right. There is another important point here, is that when we construct such a network, starting from our genes, and now we have transcription factors, and then we found a master regulator, then often this system is quite stable, and actually we often have some negative feedback loops which would compensate the activity. For instance, in many cells which get the signal for proliferation, there are some ligands like proliferatory signals which activate the whole cascade, and then cells start proliferating. But at some point, those signal has to be stopped, because the cell goes into development, and then it happens by negative feedback loops. So activity of some receptors actually goes down. But in pathological situation, it may happen that this network actually got deviated, and it's rewired, people saying, and some other molecules, some other master regulators are actually starting to hijack the system. So they take over the regulation. And moreover, they actually characterized by positive feedback loops. So those red arrows, which means the activity of that protein actually is acting on activating a gene which encode that protein. Or, for instance, in this case, this gene is really encode maybe not the end protein, but encode a component of the signal transduction where the signal has to go through. So, and it actually, also contributes to this feedback stability. And when we have such a system of positive feedback loops, then this situation gets really stuck. So you cannot get out of this autocorrelative, auto-activating loops. And that's, we believe, is often the case for many pathologies, especially for cancer, when we know that cancer cells start proliferating without any stop. And that happens because there are certain molecules which get super activated, and they hijack the whole system, and they have those feedback loops. So that's what we call so-called walking pathways, because in a healthy situation the system is rewired to the disease situation. And why it is called walking? Because what actually works, and that's really a topic of today, is the part which is here below. The whole cascade is more or less stable, although of course we see the rewiring, but the signal transduction cascades are sort of standard. But the most flexible part is the part which is here below. So which of the genes has to be activated through these by those transcription factors. So in a normal situation, in the healthy condition, those factors may activate a certain set of genes, but in a pathology, this activation profile is going away, and they start activating something else. All right, so we will jump now to the presentation. And in between, we have one question. So question is following: does the system allows for feedback from regulated molecules to regulators itself? That's exactly what I was talking about before. So that's these positive feedback loops, when the feedbacks regulated genes are feeding back to the regulators. So thank you for this very nice question. All right, so then we go online to demonstrate how this all happens. And for an example today, we will use this example of expression data. This is RNA-seq data from actually MYC-induced cells. And that's basically sort of model of oncological situation of osteosarcoma, and that's as it is discussed in this paper. So we actually took this gene expression data from this gene expression omnibus in terms of normalized counts, and let me show you how we get that. So for that, we go to our system, and on our website, you can log in using your account. And if you don't have account, you can go and register. So here in login, and it's a general login for all our tools and databases. And as I said, if you don't have your account, you register here. And of course, for the commercial parts, you have to issue send request for the price and get the license. But we have this platform part, which also we have a free part of it, so where you can register account for yourself. So once you register your account, you get your username and password, and then you log in. And here you will have actually all the listed products which you have license or you registered for. In my case, I have everything. So I have actually a product which is a combination of all our databases, TRANSFAC, this database on transcription factors and their binding sites associated with TRANSPATH, which I was talking today, the signal transduction cascade, and human PSD. This is a database about drugs and diseases. That we will not talk too much today. And then we have here geneXplain platform, which I will show mainly today. And in addition, we have actually here a separate small tool called MatchSuite. I was talking about that before. It's actually a very easy interface to run your set of genes for humans. So if you have a set of genes which are of your interest, maybe upregulated genes, you can simply put in a window here, and then it will predict binding sites in a tissue-specific manner, and it's a very smart pipeline which is running over there, so you can have a look. But today we'll go in more complex matters. So once you find those binding sites, now question, where is my master regulator? And for that we have to go for the whole suite, for the whole combination of databases, including TRANSPATH, which have information about signal transduction reactions and signal transduction pathways. Okay, let's go directly to the geneXplain platform. Actually, just a very brief outlook on this platform. If you haven't seen it before, it's actually a tool, an online tool, which allows you to work with various omics data, and not only omics, but actually many different types of biological data, genetic data. So genetic and epigenetic data, also metabolic data. And you can see here various input kind of entries in our platform. So if you have RNA-seq data, or you have, for instance, chip-seq data, or you have a list of metabolites, or you have variations. So you sequence your genome or of a patient, and you have a set of variations, or you've done your gene-wide association studies, and you have a list of variations which are associated with the disease or pathology of your interest. Then you go here, and you can also analyze microarrays if you still have them. So, and once you in such a entry point, you have a number of pipelines, and I will show a few of them, how they work. Here is your part where you put your data, and here is a set of projects, and I have quite a number of them. Actually, these ones which have the red double Y, it's actually W, sorry. These are the projects where you can see examples. So they are right protected, but where you can see examples of how this platform works on them, or even another tools. But this one is a project of myself. So the similar project will be created for you once you register your account. And then you have possibility to create in this project some folders and subfolders, and actually put your data in. So what data we are talking about? So I downloaded data from that gene expression omnibus. Let me show you the data first. And then we'll show how to upload it. So we have this example folder here, and here I have this data which I took from that gene expression omnibus. Let me open it. It's on my different screen, sorry, here. So as I said, this is a RNA-seq data. It's already pre-processed by the authors. So you have a list of actually 9,500 genes, and you have 6 conditions. And here you have actually already normalized counts. So they are already normalized. And each here, those columns which we have MIC are the samples which were treated by MIC. Well, not treated by, where they made a construct which activate the MIC. And we have 3 MIC and 3 controls. So we are going to compare this data and in order to reveal differentially expressed genes. That's a transcriptomics data. But the authors also provided actually proteomics data, which is something like this. That's here. It's a list of also around 4,600 proteins, which are shown here as a uniprot accession numbers. And here we have a value, which is already, calculated value of activity or amount of that protein in the experiment versus control. It's more or less a fold change, a logarithm of the fold change. So when it's negative, it's a downregulation of that protein. And when it's positive, it's upregulation. All right, so then we will upload these two data sets in our system. So first, let's create a subfolder. So let's call it, test one. Sorry, I have, all right, I have test one. I created it. And now to load the data, we can either go here and actually you see what kind of format can be loaded, any Excel tables or also FASTQ files or BAM files. So many types of files which can be loaded here for the expression data. You can click here and then you get these various types of loading. You can load it from your computer, or you can load it from FTP side, or from cut and paste by raw input. So we go to the computer and we select those files. As an example, I will select this RNA-seq file. And actually it detects that it's RNA-seq. And in the first column, there's an ensemble IDs. And I started the loading. So I get this file now here. So the same, we can upload the proteomics, which I did before. So here in the osteosarcoma folder, I already preloaded this data. So you see this proteomics data is here. So actually, when you load it in the system, you can sort it by clicking on this any of the column. You see those are the downregulated proteins. These are upregulated proteins. All right. So, and first what I did is actually I run Lima. And what is Lima? Lima is the tool which is for detecting differentially expressed genes. So for that, you go to the start page and you see this is detect differentially expressed genes. And you go to Lima. And here you take your gene list, like this RNA-seq. And then you say, okay, I have normalized counts. And then I have to specify what is my treatment. So this is my treatment. It's called, the experiment. And these are my controls. And then I run the Lima. So we can give a different name because I already ran it before. And then it starts analyzing my data set. I already ran the Lima before, so I can show this the results of Lima, which is here. So you get such a list of 9,500 genes, which are, you see it's a little bit smaller than input because it's filters also for those cases when there is not a significant amount of signal. So it goes also for filtering when signal is quite low. So that's, right. So, and then we can, as I said, we can sort it up or down and see which genes are up or down regulated. And also the Lima calculates the p-values and the adjusted p-values. So actually the adjusted p-values is the p-values which controls the multiple testing. All right, so from that we can actually easily find which are upregulated genes or which are downregulated genes. And so as I showed before, it's done with these filtering options. You can play here with columns and filtering options. So, and at the end, I actually selected this set of 271 genes, which have a log-fold change higher than 0.75, and which are in fact also have a adjusted p-value below 0.5. So we have just 271 significantly upregulated genes. And we have also downregulated genes, but for my experiments, I only done analyzed upregulated genes. In fact, we have a need for a background set, which we call non-changed genes. And for non-changed genes, again, by filtering, I detect those genes which have a very little change in log-fold change. It's around the 0. And we got this 1,800 non-changed genes. And this will be our background for the promoter analysis. And the promoter analysis I did using, again, I came back to the start page. And here we have analyzed regulatory regions of differentially expressed genes. And here you see this identify enriched TFBS in my promoter using TRANSFAC. And as you can see, this is a pipeline. Here below, you see this pipeline. And by the way, these pipelines can be also modified. You can modify parameters of this pipeline or add or remove different steps of it. So we'll have a separate, I hope, of coffee break of explaining how these pipelines can be analyzed or can be changed or created. So what I simply do here is I put here actually a list of upregulated genes. Where they are. So here they are, upregulated genes, and the list of non-changed genes. And by the way, if you don't have a non-changed genes, but you have just one list of genes, so by default, it's using the housekeeping genes, promoters of housekeeping genes for the control. But if you have the full experiment and later, of course, it's better to use non-changed genes from your experiment. And then you have to specify here what type of annotation you're going to use, what is your profile, which is a set of positional weight matrices. And we use here all of them, all 7,000 or more. And then there are other parameters. Important is this parameter of the length of the promoter, which we took minus 1,000 plus 100. Around start site, and that's right. So then we can basically run this workflow, and as you can see, when it runs, it goes through this pipelines and it shows which step in the pipelines are working at the moment. So it will take some time, but I of course did it before. So these are the results of that pipeline run. And the important part of it is this enriched motifs. And it gives these positional weight matrices which are enriched in my set. And it gives a lot of parameters to show what is enrichment. And actually, the most important thing is this site FDR, which shows how this enrichment, which means what is enrichment? It shows how many sites were found in the promoters of upregulated genes and compared it to the sites, number of sites found in the background set. And then actually it estimates whether the difference, this ratio between them, which is is maybe not very high, like 0.2, but it's very significant because number of sites is very high. There are also a number of other parameters here, which you also controls whether those sites are equally distributed in my promoters or they are more focused in subset of a group of genes. So that's done by this sequence p-value. So I have actually a question in my Q&A. What is a good measure to decide about differentially expressed genes, log-fold change higher than 2, 1.5? All right, so that's a very good question. I think I will keep this question for for the future coffee breaks, because it's a very big question. Here, as you see, I used actually a cutoff 1.5, which is a standard in many publications. But and I combine it with a p-value cutoff. And the important is not to be too strong, too sharp in your cutoffs, because otherwise you will get just very few differentially expressed genes, and you will not do anything with that, and that's biologically not reasonable. And as I said, I will talk about that in the future coffee breaks. All right, so you see now that's results of my search, and as you can see, it gives a nice also produced a nice report which you can also download as PDF. But we go further to see the results. So, and actually what we see here, we also see the sites where they're located, and I showed it before. So you can really investigate where those sites are located and so forth. But the topic of today, again, is rather the transcription factors. So we found the binding sites and we found the motifs which are enriched. But now what are the transcription factors? And here is a list of transcription factors. We have 175 transcription factors, which are associated with these positional weight matrices. And here we have this yes to no ratio, which is enrichment of those of these binding sites and the promoters of the genes, of upregulated genes, for the motifs which for this factor. And at the top you see this zinc finger factor 433, and the enrichment is actually 6 times, although the number of sites is pretty low here. And this factor AP2 is actually enrichment is very high, and number of sites is very high, and enrichment is very high, and the p-value is very good. So it shows really that these binding factors are important. Of course, we can filter them more by the various parameters to be sure that our factors are really, significant. And I did filter them further, actually, let me see, by, yeah, I did filter them. Where did I filter them? All right, so I actually filtered them here. I start filtering starting from this motif list, which I showed, and I required that the p-value should be very strong, and not only for the site FDR, but also for the sequence FDR. So at the end, I came up with this list of 79 genes encoding these transcription factors. So I already converted them into the genes, and some of the factors you can see, they may be found by many motifs, by many different positional weight matrices. And I also annotated those genes by this fold change and p-value from the transcriptomics experiments. So you see, for instance, MYC itself. Of course, that's experiment with MYC. It's very highly upregulated. And the motif for the MYC in the promoter of the gene is significant. You see, this is a values for the motif enrichment, and this is actually proteomics data, which I also appended in this table. And as you can see, on the proteomics data, the MYC as a protein is not actually changed a lot. It's 0. So that's sometimes this discordance with transcriptomics proteomics happens. But we should actually use this, in our analysis when we combine these two. So, but if you sort by proteomics, so you see there are proteins which have highly upregulated on the protein level and also on RNA-seq level a little bit. So, for instance, the MAX, which is actually a MYC association factor. So it makes a lot of sense that the other component of that complex is upregulated. So we know now which factors are involved in regulating of my genes. Now we have to go upstream from them. And to go upstream, we have this tool in our method section. So we go to the method section. and we go to the molecular network, and we actually here you see a number of tools to work with molecular networks, and the tool which is which we want to use here is a regulator search, and this tool is exactly doing what I showed on my slides. So it starts from the transcription factors, and it goes upstream in the signal transduction cascade of a database, of TRANSPATH database, and finds the master regulators. And this tool has various options. And let me show you the options which I choose here. So actually it's here. Let me see. Downstream, so proteomics, no, let me see where I did it. All right, it's here. It's here. All right. So that's already a result of that analysis, but first I want to show you the options which I use to run this analysis. And actually in our platform, it's very nice in the sense. So each time you see the results of some analysis tool, here in the info box, you see how it was run. So you have the history. So here we run the regulatory search. And if you click here on it, it will open here with all the parameters which were used to run this regulator search. And as you can see what we did here, we put here the list of proteins, which we found, these are the, actually, that's another point which I have to emphasize here. You remember we got in fact, we got 79 genes encoding transcription factors, and then I filtered them further by, I require that those genes has to be expressed in that system at least. So if there is no expression detected, so maybe it's not acting. So that I got actually 37 genes which encode for transcription factors, and then I converted them into TRANSPATH IDs. So there is a conversion tool here which allows you to convert from one database to another. And now, after such a conversion, I have this regulator search tool here, and I put here this list of 37 transcription factors, and then I require the radius 10, which means it goes upstream maximum 10 steps. As you remember, these are many steps, like receptors should activate kinase, another kinase. So those steps usually are quite short, not so long. So to be sure that I took to the very far, I put just 10. So it's a kind of a very far away from transcription factors. There is some score cutoff, which also present in that formula, which I showed in my presentation. And then there are two important, one important thing here is that we can put so-called decorator. It's a strange name though, but it's named decorator. So which means this network of signal transduction can be changed before we do the master regulator search. And we do it by putting the context. It's called apply context. And that's exactly what I showed in my slides when I put the proteomics data on the network. So I know which proteins are actually upregulated, which proteins are downregulated. And I put here the context table of those proteins which came from proteomics experiment. And I choose here the apply context. And then when I run this, I got this list of master regulators. Well, actually, it's a list of regulators. Which ones are really master is depends on the score which we want to define. So here at the top we have the maximum score, and you see it's the top score is gets proteasome, which is quite often the case that the proteasome is involved in many processes. So maybe it's not as specific as we would like, but it's pretty specific for this set of transcription factors which we put here. So out of 37 factors, it reaches 19 from them. And then, we have this master regulator. So the second is ERK2 kinase is another master regulator. And we, of course, can see that on the screen. So by selecting those master regulators which we are interested, and visualizing them here by this button. So it's now starting the drawing this cascade from the master regulator, and here you see this network, which is predicted for the master regulators. So that's the way from transcription factors to the pathways. So actually, it's a way from transcription factors to the master regulators, and which is constructed by our algorithm. And you see these master regulators are at the top, and these transcription factors, which are blue here, are at the bottom. And you see intermediate molecules which connects those master regulators to the transcription factors. And basically what you also can do, you can put here the expression data on that network, for instance, like this. I took this here, right? So it actually gives. I can give a bit more size of this marking. So actually, what you see here, those proteins which are upregulated, they have this red aura, and those which are downregulated, which have this blue aura. So those which have very little change, they have very little shade. So you can actually vary with this coloring. All right, so that's how it works. The last very small point here is, which I mentioned, that with this network, you can do quite a number of things. For instance, you can do various layouts here. So this is a hierarchical layout, so you can maybe do it also in horizontal way. So when your master regulators are at the left and the transcription factors are at the right, there are other few actually options to do the layout. And then after layouting, you can save this in your computer in several various formats. For instance, you can save it as a PNG picture simply, and then you can put it in your presentation or in your paper, or you can also increase the resolution of that. Also, what I also mentioned, you can to work with other tools. You can save this in standard formats. For instance, there is a graphic ML format for a number of tools which are using it. Also, there is a Cytoscape format, and actually there is a generic format which many tools are using. It's SBML format, which is systems biology markup language, and you can save it as a file for this tool. And if you have these tools, for instance, here I have, let me find my tool. I have opened this Cytoscape. So we can upload this network into Cytoscape. So here you see a Cytoscape screen. So actually I will open my network. So here it is. Actually, I have to import it rather than open. Import from file. Import from network from file. It's from SBML format. So the Cytoscape is imported this network, and you see it doesn't apply any layout because SBML format doesn't contain any layout inside. So we have to apply the layout inside Cytoscape. And in Cytoscape, there are a number of layout tools and much more than we have in GeneXplain platform. Various types of layout. For instance, this nice layout, which is a circular layout. So you see it's actually layouting all our molecules and reactions into a circular way. So, and actually what you see here is a connection between molecules, and in between node, you see the information about the reaction. So, and you can apply also other types of layout and do other types of analysis using Cytoscape, and you can combine different networks together and also save them in various formats. So the Cytoscape is a very useful tool to apply many algorithms after what we did in geneXplain platform. All right, so that's what I wanted to talk today. And it took me a little bit longer than I actually anticipated. But now I would like to have a pause a little bit for your questions. All right. So one question was, actually recently it came, whether these networks and the search can, whether we can, the user can introduce own interaction data. That's a very important question because, of course, if you look at the databases, there are several databases of pathways and networks. And here I showed you the usage of the network database from our TRANSPATH database. So we have this TRANSPATH database in our list of databases, and we have here, as I said, more than 1 million reactions in this database. But there are other databases of the networks. For instance, there's a REACTOME, or there's also, you can see here databases like Recon. This is a database of metabolic reactions and metabolic pathways. And also there is another database which we have in the database list, which is also open for everybody. It's called it's called GeneWays. Let me find it. It's here. It's GeneWays. It's a very interesting database. It collects reactions from literature using text mining tools. And this is it was created by analyzing automatically more than 1 million different references. So it's a huge database of reactions. And in such a case, we have various databases, and my tool for the regulator search, it includes only TRANSPATH reactions by default. And the question was whether we can use other databases. Right, so first, of course, we have here, you can select a database of your interest here among the, it's called, search collection. Default is TRANSPATH, but you can go use GeneWays, for instance, and then you actually goes in completely different network and do your work in another one. Or you search a REACTOME database, so which include reactions extracted from REACTOME. But the question was actually whether you can mix different databases, and you indeed can. And here I actually did that for you before, because I was preparing answer to that question. There was this proteomics data shows that some proteins are very much upregulated by, unfortunately, they were not found by our regulator search, because there is no reactions in TRANSPATH from those molecules. But those reactions are actually present in GeneWays. So, for instance, I found here such a network which from GeneWays, which connects this protein, which is SDC1, which is highly upregulated in my experiment, but it was not found because TRANSPATH doesn't have those reactions. So they have, but GeneWays have, and that's why I want to add those reactions into the search. And for doing that, I have to prepare those reactions in extra file, and that's an extra file which is here. Let me see, is it that one? yet here. So which shows those connections which I want to add. So I want to add 36 connections from various proteins, which I found in my experiment highly upregulated, to the other proteins which I extracted from GeneWays database. And it's like from protein to protein. Well, it's actually using a gene symbol for that. And that's kind of colon saying it's activation or inhibition. And then also the same is done. I converted it into accession number from TRANSPATH. And when I did such a search using these additional networks, I actually found this network. I found this master regulator SDC1, and this reaction, which is blue here, is added to my search from this collection. So this is additional reaction which was not present in TRANSPATH, but adding this reaction, we can find this master regulator. So that's a way how you add these reactions. And actually, the details how we adding these reactions to the regulator search, it's again by decorators. You actually have to add a new decorator, which is you see add interactions, and then you're adding here these new interactions. You actually can add several sets of interactions from the various databases, and then it will improve your search. Okay, so in the meantime, we got 2 other questions. So if you are not yet both, I can answer them. The question is the following: is it possible to construct the master regulator-TF network without proteomic content? Are there any reconstruction example of this type of network for microorganisms? Okay, the first question is yes, indeed. So just in this example, I showed the case when we have this context, and we can add this context to the of the proteomics experiment, and this context give additional weights to the network. But if you don't have it, we simply don't have this data. So we remove it. We don't have any decorator, and then the search is done just purely on the TRANSPATH database without any additional data. It takes TRANSPATH as it is, and those connections are used basically to from a TRANSPATH database without additional weights. And often we do this because not very often we have proteomics data from the same experiment, and very often we have just transcriptomics data, and that's why this is a usual way how we use this upstream analysis. Concerning the microorganisms, that's a very important question. It's very little knowledge about signal transduction networks in microorganisms, and basically also in general, this tool here is mainly focused on eukaryotic organisms, and you see those genomes which we use here is a human, mouse, rat. We have a few model organisms like nematode, Saccharomyces cerevisiae. Actually, and we have one sort of a microorganism. It's not, of course, microorganism, but it's a single cell. And then we have zebrafish and Arabidopsis from the plant domain. And for the bacteria, unfortunately, we don't have much data here for analysis of bacterial promoters or for analysis of bacterial networks. All right, so another question: how can we plot the signaling pathways for TB host hub? TRANSPATH, I think, host hub genes, or any genes using GeneXplain platform. How can we plot the signaling pathways? Actually, I don't understand what is TB host hub genes. Is it tuberculosis? I'm not sure what is TB. I'm sorry, but signaling pathways using GeneXplain platform. All right, so I don't know, maybe one answer would be to, we can directly plot signaling pathways from the list of genes, maybe that's what is your question. So for instance, if you have a list of genes, like these upregulated genes, which we had here, we have these upregulated genes, right? And we simply want to know to which signaling pathways these genes are mapping to. Then we use tools called, here it's called functional analysis. And that's a tool. There are two tools. One is called enrichment. Indeed, tuberculosis genes. All right. Okay, but let me finish what I suggested. So this enrichment analysis tool is what it's doing is you take a list of genes, you put it here like this, and then you select here a database of canonical pathways. For instance, these TRANSPATH pathways, and here it is mapping not to the whole network, but to those canonical pathways. And I talked about that before. We also put quite a bit of efforts on constructing canonical pathways. So those standard pathways which are functioning in many different cells. And you can simply map and find the enrichment, which I did. Actually, I have it here. This enrichment by pathways. So, for instance, this list of genes is it enriched this small pathway, which is from gene VAF to NF-kappa-B, or it's enriched in bigger pathways like EDAR pathway. And here you can see how those genes are actually, maybe you're familiar with this gene set enrichment plot, where you have we have our genes here on the X plot, and here we see this sort of Kolmogorov-Smirnov score, and this red plot shows each peak here is one gene from this EDAR pathway, and we see enrichment if many genes from EDAR pathway is among the list of our top regulators. So in fact, in this case, there are not so many, although in the middle it goes up. But if you sort this by rank at max, and we see, for instance, this network, so you see it's in the beginning, it's quite a good enrichment. So you can plot that. And in fact, you can also see this network, and you see how your genes are mapped to this network. It shows here. So this is a network, not network, the pathway. And those which ones are colored here are the genes from my set. So you see there are a number of them. Well, actually, the different colors means different expression mappings. So if you select a log-fold change, you see them just one color. This is upregulated proteins, actually, and these are downregulated. And you see they are mapping to this pathway. All right, so that's coming back to tuberculosis. Tuberculosis hub genes, you mean in the, I guess, in the microbe, in tuberculosis microbes. As I said, we don't have any data about microbial analysis, but in terms of tuberculosis as a disease, there are, of course, on the host side, you can analyze a lot on those genes which are genes of sensitivity to tuberculosis, and we have actually a couple of, we have one paper in collaboration with in tuberculosis research, when we analyzed networks in the host, in human, of sensitivity to tuberculosis. All right. Okay, one more question. Is it possible to discover new regulatory pathways based on expression data? Right, that's a very good question. That's exactly what I showed to you. So those networks like this one is in fact a new regulatory pathways. As you remember in the beginning, I talked about rewiring. So, and very often we have this paradigm of canonical pathways like AGF pathway, TGF beta pathway, or those canonical pathways which are sort of canonical. So they're fixed picture, and we believe they are functioning in any cell. But we know that pathways are very flexible, and depending on conditions, they are rewiring. And for instance, such a pathway from this protein to these transcription factors is a context specific. It's we believe so through our reconstruction, we believe this pathway is actually functioning in that particular cells upon activation of MYC. So in that very specific condition, this network is rewired towards this pathway. So in that sense, you can say this is a new biological pathway, which is specific for that particular condition. All right, I hope I answered that question. All right, so I don't see any more questions in my Q&A list, but okay, so we have already more than 1 hour on our coffee break. So our coffee is already cold. I will finish soon. But please send us questions. You can send it just by email, or there is a form of registration for the next coffee breaks. You can register for the next coffee breaks and put your questions over there. So, and we'll come to those questions in the coffee breaks, and we'll try to answer them all. Of course, I'm sorry today my presentation took so long because it's a really complicated topic, and we didn't have much time for answering questions, also those ones which we accumulated during our time between coffee breaks. I hope next coffee break we will spend more time for answering your questions. Okay, thank you. Okay, I have some question in the chat. Yes, right. So there is in the chat, you see there is a link where you can ask questions, and we will address them next time. All right. Thank you very much for your attention, and thank you that you are coming to our coffee break. And take care, and I will eat my apple. Thank you very much. See you.